Hello, everyone, and welcome to today's webinar, Unlocking Insights in Complex and Dense Neuron Images Guided by AI. My name is Choi, Product Owner and Manager for Data Analysis at Leica Microsystems, responsible for the Avia product line, and I'll be your moderator for today's event. Today's educational web webinar is presented by Labroots and brought to you by Leica Microsystems. To learn more, visit www.leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd like to now welcome our speaker today. Tran is the Global Business Excellence Manager for the Data and Analysis Group, responsible for the commercial aspect of Avia at Leica Microsystems. Quyen received her Ph.D. in biomedical engineering at the University of Wisconsin-Madison, where she examined stem cell behavior in 3D protein-based constructs fabricated using multiphoton microscopy. Quyen has worked with Avia since its launch, utilizing her perspective as a scientist and Avia expertise to bring AI-powered image analysis to help researchers propel their work forward. Quyen, you may now begin your presentation.
Hello, everybody, and welcome to the webinar. My name is Tran. I am the Global Business Excellence Manager for the Data and Analysis team at Leica Microsystems. Today, I'll be introducing you to our latest release, Avia 12, in which we introduce tools that help you unlock insights in complex and dense neuron images. A brief look at the history of Avia for those who are unfamiliar with our software. Avia started in 2017 with Avia 5 as it was built on a pre-existing software. Since then, we've had a number of releases that introduce tools for neuroscientists, cell biologists, cancer biologists, and developmental biologists. We've also incorporated several AI components, such as the pixel classifier, object classifier, and deep learning into Avia. Today, I'll be introducing you to a few more tools in Avia. Now, we know that neuroscientists use a broad range of imaging modality, such as fluorescent-based or Golgi-stained images. And they're trying to capture neurons in a wide range of different environments, including environments that have sparse versus dense neuron networks. This slide is just showcasing a few examples of the type of images and complexities they're working with today. This range in both methodology and networks can make it particularly hard to analyze and understand the neural morphology and biology. So today with Avia 12, we are adding a few more tools to your toolbox to help you reduce this complexity and to gather additional insights. Here's a brief look at the key features of Avia 12. We're introducing soma detection powered by AI. We've also expanded our 3D neuron analysis recipe to help you tackle challenging data sets and to speed up your analysis. Avia 12 is also introducing new tools that help you create insight and a few other AI updates and tools for you to utilize in Avia. Let's delve into the soma detection powered by AI. This is a new option in our 3D neuron analysis recipe designed to help you better detect somas in dense neuron images. You can see here a quick snapshot of the soma detected using our non-AI versus our AI-powered methodology. With AI, we were able to detect additional somas in this dense network, as indicated by the red arrows in the zoomed-in portion of the image you see here. We compared these two methods across multiple images and quantified this improvement by examining the recall score. Recall is the total number of true positives divided by the total number of true positives plus the total number of true negatives to get us an estimate of the improvement. In the 4 images I've shown here and additional images that we've tested on, we saw improvements in the recall value when AI was used to detect the somas. Additionally, this AI method also allowed us to partition the soma better, especially in areas where there are soma clusters, as illustrated by this particular image. Now, how does all of this come together in AVIA? Essentially, we're bringing the cell-pose deep learning model into the soma detection step. Once you select this, AVIA utilizes two new parameters to perform the detection. The seed detection intensity threshold is the intensity that cell-pose is going to use to detect the seed point or the starting location of that detection. The cell probability threshold examines the output of the cell-pose deep learning model and sets the threshold of where to detect the actual soma. You might also notice here that the two previous soma detection options are still available to you. We have retained these two options as they are still great to use for low density images, and we still highly recommend these two options for those situations. Now let's bring all of this together with two example videos. The first is utilizing this new option of soma detection by cell pose to detect neurons in a dense neuronal network. The second video here is a closer look at that partitioning capabilities that I mentioned earlier. Now, let's look at the next set of key features for AVIA, which is the expanded flexibility of our 3D neuron analysis recipe. There are two updates here. First, we have separated the channel input for soma and dendrite. This allows you to utilize the most optimal channels you have for these two critical components. It also allows you to bundle this with the pixel classifier output, which I'll show you a little bit more in our demo later on. If you only have one channel for both soma and dendrite, don't worry, that's not a problem. You can simply select the same input channel for both of these for both the soma and the dendrite. We've also added a preview functionality for the soma detection step. Once activated, the somas locations are shown as spheres, and this will allow you to optimize the parameter for the soma detection step before you start to detect the dendrites and the spines of the neuron. This next example video here showcases neuron tracing results we were able to obtain from a 3D Golgi stained image using these two updates. Now, how did we achieve this? A brief look. I'll show you more in the demo later. First, we utilize AVIA's pixel classifier to enhance the somas and the dendrites separately, as they have very different characteristics. We also made sure in the pixel classifier to enhance the in-focus regions of these two components. We then used the new 3D neuron analysis recipe, taking advantage of the two new channel input functionality to trace the neurons you see here. Now, our next set of features are new tools for insight generation. In this example, we are looking at meshes that are color-coded based on its proximity to the neuron dendrites. How do we create this video? By utilizing a new feature, 3D spatial measurement in AVIA 12. This feature is an expansion of the AVIA object relational tool. And in this tool, 3D relationships can be established between any two object groups, whether you're establishing a relationship between meshes and meshes, neurons and neurons, or neurons and meshes. These relationships are then used to measure and calculate certain things. It's also important here to note that these measurements are calculated by looking at the surface to surface and not the center to center, which is critical when we are examining and measuring distance with objects that are complex or have non-globular shapes. You can imagine here the distance between the center of a dendrite and a mesh may be very different than calculating the distance between the closest surface of a dendrite and the surface of the mesh. This feature also comes with pre-established relationships and measurements for your convenience, including the detection and distance calculation of the nearest object or the nearest 5 objects, as well as counting the number of objects within a certain distance or overlapping objects. In AVIA 12, we also implemented 3D shoal analysis for researchers looking to quantify their neuron morphologies. This slide shows just a brief snapshot of the different types of morphologies we saw in one particular image, which you can now quantify using 3D shoal analysis. This analysis is also another expansion of our 3D object relation tool. Once launched, the analysis is immediately performed on your first neuron object set and using default shell count and radius steps. These three components can of course be modified to meet your needs. We've also implemented the ability to view this analysis as a heat map and to be able to export these values for further analysis if you so wish. But it doesn't stop there. AVIA 12 comes with new charts and charting features. First up is the selection by polygon within our scatter plot. Using this feature, you can select any group of objects that you want in any shape that you want. Once the selection has been made, you can create subgroups for further examination. In my particular example here, I looked at the scatter plot of the XY coordinates of cells within an organoid. Using the select by polygon feature, I selected and separate the interior and exterior cells. Of course, this is not the only way to look at the cells and to separate the cells, and you can probably already start to imagine numerous other ways that you can separate and organize the cells that would be interesting to you. Second item here is our new violin plot. So you can examine the distribution of a particular measurement across different groups or subgroups. My example here, I've taken those two subgroups I created above, the exterior and interior cells, and looked at the distribution of the number of contained vesicles in each cell. Additionally, this new plot automatically calculates the mean and standard deviation for each group and also performs a Welch's t-test between each group. These values can be seen when you hover over the chart here. Third, we've added a Pearson heat map to your chart options. This chart displays the Pearson correlation coefficient in a heat map style for all of the measurements in a particular group. With these new features and charts, we hope to provide you with the tools you need to help you connect your analysis to insights into the biology of your neurons, your cells, or other objects of interest. Now, our last group of updates here. Our first update is a connection to the Bioimage Zoo. If you haven't heard of it yet, the Bioimage Model Zoo is a community-driven effort to make pre-trained deep learning models more accessible to the community. This is just a quick snapshot of the models within the zoo. In AVIA 12, we are hoping to help in this effort by bringing 4 of these models to you directly. We have repackaged these models so that they work directly in AVIA's recipe console. As shown in the video you're watching now, you can simply drag and drop the models to work within AVIA. No additional setup is required for you. The 4 models we have worked with are the HPA cell segmentation, the HPA nucleus segmentation, the bacterial segmentation, and the pancreatic model cell phase, the pancreatic cell phase model the pancreatic cell phase segmentation model. Apologies for that. These models are now available in AVIA's Deep Learning Model Library, which you can download and start to use immediately. We want to express our thanks to the organizers and leaders of the Bioimage Model Zoo in making deep learning easier for the scientific community to use. We also want to send a special thank you to the contributors of these 4 particular models that we've incorporated into AVIA. You can, of course, find out more information on the Bioimage Model Zoo on their website, which I've included here. I've included a link here for you. Another update in AVIA 12 deals with AVIA that learns. For those who are unfamiliar, AVIA that learns was introduced in AVIA 10 for 3D object analysis. Essentially, you click a button AVIA then examines your image and using our AVIA that learns deep learning model predicts the recipe parameters for you. This essentially provides you with a starting point for your detection. In AVIA 10.5, we expanded this feature to 3D neuron analysis. And with this release, we have updated the deep learning model to better predict these parameters for you in your 3D object analysis. In this example here, we have a very challenging data set. Cell clusters are seen in the edges of the organoid. The organoid also contains cells with high and low fluorescence levels, cells of different sizes. With our new AVIA that learns model, the parameter prediction for this particular data set and other data sets have improved. So it is a much better starting point for you to create your analysis results. Last but not least, we have updated our region of interest tool to help you better analyze what's crucial to you. In AVIA 12, we're introducing a region of interest or ROI manager. This lets you see all of your ROI in one place. You can add more 3D ROI boxes. You can draw a freehand 2D ROI. The manager also gives you the exact XYZ dimension as well as the size of your ROI. And you can input and type in the values here to modify the ROIs instead of using the spheres that was done before. Additionally, you can select to view a specific ROI, you can crop that ROI, you can even control whether the ROI is going to be used in the recipe or the pixel classifier. Essentially, this is a one-stop shop for all of your region of interest needs. I also wanted to include a special note to our current AVIA subscribers. Those in the AVIA Elevate Neuro and the AVIA Apex package can update their AVIA version to AVIA 12 and immediately get access to all of the features I talked about today. For those in the AVIA Go and the AVIA Elevate Cell package, don't worry, you are still receiving a number of these updates. I showed them in this presentation in the context of neuroscience, but many of these features are still a part of your package. This includes our 3D spatial measurements, those new violin plots, the Pearson heat map, the ability to select by polygon, to directly apply those 4 deep learning models for bioimage zoo, the update to AVIA that learns, and also the ROI manager that we just looked at now. So with that, I would like to transition us to the demo portion of this presentation. In this example dataset, we can see many challenges for neuron detection. First, this is a dense dataset with a lot of somas and dendrites. Second, there are bright intensity somas as well as low intensity somas within the same image. Both of these features make it difficult to analyze neurons with intensity-based algorithm. So I'm going to demo how to use our new soma detection powered by the cell post deep learning model. We're going to use our current 3D neuron analysis FL recipe. Let me expand the soma detection portion here and show you where to select the option to detect soma by cell pose. Since this is a dense data set, let's go to the ROI manager and create an ROI. I've already created one. Let's select it and clip the view so we only see this portion of the data. I'm also going to put it more centered so we can see it a little bit better. I'm actually going to go in and modify the Z size here so that we can focus more on the soma detection portion with just some of the dendrites in this particular step. I'm also going to select the apply to option so that the recipe only applies to this ROI. You'll notice that this will also select this option in the recipe console. Let's expand the dendrite section so that we see everything we need to work on. In this case, I'm going to take advantage of Avia that learns for Neuron and have Avia help me find some starting parameters. Once click, Avia will examine the image and provide me with starting parameters for this neuron detection. Because I know this data set, the soma diameter range seems large, so I'm going to go ahead and modify this to the appropriate size. I'm also going to make sure that the cell pose option is the one that is selected. Let's take advantage of another feature here, Soma Preview. Let's toggle the channel on and off here so we can see the preview better. Looks like we're missing some soma, so let's decrease this seed detection parameter a little bit. That looks better. Now let's run the recipe to see the complete neuron tracing on this particular region of interest. The soma detection looks pretty good here, but I'm seeing maybe not as many dendrites as I would expect. So perhaps let's lower that intensity threshold for the dendrite detection and run it again. So this looks a little bit better. And we can continue to adjust these parameters to get the best results. But maybe for the sake of time here, I'm going to show you some analysis I ran earlier on the whole image. Now, using our new AI soma detection option, we were able to analyze this rather complex data set. Now let's take a look at the charts to explore these results further. I'm going to change the axis of the scatter plot here to total intensity and soma volume. I'm also going to deselect these other groups so we can focus on the somas detected in the whole image. Now I'm going to use the selection by polygon and select somas with high intensity and large volume. I'm going to now separate this into a subgroup, and I'm naming it simply large soma high intensity. Let's remove these soma so I can do the next selection step a little bit easier. Now I'm going to select everything else, all of the other remaining somas, and make a new subgroup out of this. This time I'm going to call it smaller soma, lower intensity. I'm going to now add everything back in so we can see a quick look. And then transition to our new violin plots and look at the dendrite count for these two subgroups. Let's expand this a little bit a little bit larger so that we can see this better. You can see here the difference in distribution of this measurement in the two subgroups. And hovering over each group, I can also view the mean, standard deviation, and the significance between these two groups using the Welch's t-test. I'll stop exploring this data set for now, but you can see that with these new charting features, you can begin to generate insights with the analysis result. Let's check out the next demo data set. For those who utilize this technique, you will instantly recognize this as a Golgi stain neuron. Our first step here is to utilize the pixel classifier. I'm going to create a pixel classifier specifically for the soma. I'm going to change this background color real quick so we can see it a little bit easier instead of the gray on gray. I'm also going to go ahead and select a large kernel size because I know that the somas are large in size. So I want to make sure everything is captured for the pixel classifier. Now I'm going to paint in some examples. This is my technique here that you'll see. I usually outlined an area of background first and then go in and paint in the region of interest. You don't necessarily have to do it this way, but this is my preferred method in doing this. So paint both a in focus and a region of the in focus somas here. I'm also going to provide examples of dendrites and a general background within the image so that the AVIA knows this is also not something I want to detect in this particular pixel classifier. Now let's teach this pixel classifier. And then apply it to this particular time frame and Z depth. So we have a quick look at what it looks like. Now, for the sake of time again, I'm going to show you some pixel classifier results that I ran earlier for the soma. Now let's go create the pixel classifier for our dendrites. So renaming this to dendrite and recolor coding that background option again. I'm also going to the feature section and click the line option as I want to use the correct teaching features to optimize for line enhancement. Now again, let's paint some examples of dendrites, particularly those dendrites that are in focus areas. Again, my technique here, background section first, and then going back in to paint over the specific pixels of the object that I want, in this case, the dendrite. I'm now using the paintbrush instead of the region lasso tool. And I'm going to change the size of the paintbrush here since this is a thicker dendrite. I'm also going to provide AVIA with some examples of somas so that AVIA knows this is not what I'm looking for in this pixel classifier. and teach. And then apply to the Z frame again. This definitely could use a little bit more training, which is easy. We just paint more examples. But again, for the sake of time, let's take a look at what I've done previously. Now, let's use these pixel classifier results to perform our neuron tracing. We're going to go back to our recipe. This time, we're actually going to expand the input and output section and change it to our pixel classifier results for the soma and then the dendrite. Let's get an ROI started here like we did previously. Oh, I added another ROI. Let me clean this up a little bit and remove one of the ROIs here. Again, you can do all of this from the ROI manager here. I'm going to expand this ROI a little bit so we capture the full soma here. And also change this Z location and depth to capture the full depth of the image. Let's put this in 3D and then clip so that we can look at this particular neuron. Now let's do this in a more manual process, as I know some of you probably are wondering how to do this without Avia that learns. So first, let's go into the annotation tab and do a few measurements here, looking at measuring the soma diameter and the dendrite diameter, and also perhaps a bit of the dendrite length. Let's use these values to fill out the soma diameter and aspect ratio. I like to give myself a little bit of wiggle room here. And since this is a relatively sparse data set with somas far apart, let's do the skip soma partitioning option here. Now for the dendrite portion, let's use those measurements we just did. And then zoom in to look at the intensity values. I want to take a look at particularly at the dim dendrite sections. You can see the intensity values at the bottom of Avia. So giving myself again a little bit of wiggle room here. And then remembering to select the apply to option and then run the recipe so that we only look at this section. While this is running, I should note why we created two different pixel classifiers for this data set. This mostly lies in the difference in the morphologies of the soma and the dendrite. While we could have created one pixel classifier, it would have required lots of training, and doing them separately allows us to better fine-tune the features to match the morphologies of these two cellular components. You also see here that as I was talking, I launched this into our 3D viewing mode here, as the neuron results can only be seen in 3D. All right, so our analysis is done. It did a decent job, but of course you can continue to further fine tune this if you would like. I'm gonna go ahead though and do my thing of providing you with previous results that I've done, and this time for the full image. Let's remove this clipping here. Zoom out a little bit so that you see it a little bit better. And then finally, let's turn on those results that I have already done. This looks pretty good. and we can see that there are multiple somas with dendrites connected here. Now let's go to the final demo data set here. In this one, we are going to focus on exploring the analysis results and not necessarily generating them. So I'm launching this into 3D, and we're going to go into the relationship spreadsheet and establish a relationship. I want to take essentially a look at the meshes. And find the nearest dendrite set. I want to optimize this for accuracy and also to calculate the default measurements here. All right, we need to set this left-hand side as our primary object set. So both of these need to be object-based. And then let's select a particular mesh. You can see the nearest dendrite here is about 23 microns away. And on the right-hand side, you can see exactly which dendrite segment it's correlated with. I select all and then zoomed into this section so we can see a little bit better. Dendrite segment, neuron mesh. Let's take a look at another one. And select it all again. You can see here, this is a closer mesh and dendrite connection, which is correlated here in the detection measurement. We can also change the view here in the color coding to take a look at this relationship in the broader scheme of this entire image. Let me zoom out a little bit here so you can see a little bit better. So see, you can see here the blue indicates meshes that are close to the dendrite, while as we get warmer and more towards the red, it is further away from any dendrite segments. Now let's do another analysis. We're going to do shoal analysis this time. We're going to go into our 3D relationship spreadsheet again and select the shoal analysis. This is again immediately launched and with the default values being used to generate the analysis. All right, let's run this analysis but with more shells and smaller radius so we can delve into this a little bit better. Refresh this here and expand it so we can see it a little bit more. You can see here with the heat map, we can see most neurons have at least one relatively long dendrite, with some dendrites having multiple long neurons. With that, I would like to conclude the demo portion of our presentation here and thank everybody for coming to our webinar. I hope you found that the tools we introduced today are exciting and useful for your work.
Thank you very much, Quyen. We have a number of questions here for you, so let me start with the first one. The first question is, can you measure the distance between objects in time-lapse images?
Yes, so the object relationship tool, the distance measurements we were looking at and things like that also work for 4D time-lapse images. So that is possible in those kinds of data sets.
Thank you very much. For those of you who may be joining late, please enter your questions in the Ask a Question box located on the far left side of your screen. We'll answer as many questions as we have time for. Next question we have, I do believe you showed it but we're getting a question specifically about whether you can measure the distance between different types of objects, such as dendrites and cells.
Yes. So I did mention this, but let's talk about it a little bit more. So the types of objects that you can measure the distance and that relationship tool is set up for is really broad. So we can do, neurons to neurons. If you have two different types of neurons you're looking for, the example I showed specifically was neurons to a particular nucleus. So we're looking at neurons to meshes. We can do neurons to cells, neurons to vesicles, cells to cells. All of those combinations are there and provided for you. The other thing to kind of mention here, especially for the neuroscientist or anybody who's looking at potentially really unique cell shapes or cell shapes that are not round and globular here, is that we're doing specifically those distance measurements surface to surface. So if we have, you imagine kind of a tube of a dendrite here, if we take the center of that and measure it to a center of a different object, it might be different if it's on one end of the dendrite. Whereas if you're measuring the surface, those types of measurements are going to be similar to what you would expect, and that we're measuring the actual distance between those two objects and not the center to center. So that's an important thing to keep in mind, especially if you're working with neurons or even muscle cells, which tend to be a little bit more elongated.
Thank you very much, Quyen. We have a lot of questions here, so let me pick a few. Can you export the Pearson correlation coefficient chart with the values that are generated?
Yep. So in the chart option, the area where the chart is, there is an ability to export that kind of data out. Also, don't forget, you can also export out the values of the measurements overall within the spreadsheet also. So two ways to export out the data and for you to do further analysis if you want.
Next up, the question is about whether the new version is still compatible with the Zeiss image format.
Great question. So Avia is essentially imaging platform diagnostic. We have optimized it with the Leica system and everything like that, since we are part of Leica. But you can use CZI files, list files, anything tip related, things like that. So it's imaging platform diagnostic. So for sure, Zeiss files will work.
The question is, can you just, do you need to do any special conversion for Zeiss file formats, or can you just open that with an Avia?
Yep, good question. So yes, you can just kind of drag and drop or go file open and open the CZI files. We do still, however, suggest that you convert them to Avia TIFF. Again, this is more of an optimization so that the next time you open it, especially for those large 3D data sets, the multi-resolution and multi-tier file format that we have for Avia TIFF is going to allow you to see things better and to analyze things a little bit faster. So it's just a little bit more optimized. But if you're just looking at the file and want to see your images, a simple drag and drop will still accomplish that for you.
Fantastic, thank you. Next, can you do a batch analysis?
Yep, so batch analysis is possible. I didn't showcase that here, but yes, so batch analysis is a functionality. Essentially, you can set it up in terms of using the recipes that we have viewed here. So for example, let's say you go in, you use Avia that learns and optimize your parameters for neuron analysis, and you have 20, 30 images that you need to process through. That's possible through our batch analysis. You can also add layers on top of that too. So the example I could give here, let's go back to the demo of the Golgi stain, right? We created those 2 pixel classifiers and then we ran the neuron analysis recipe. You can pair that together and batch it that way too. So it's got a lot of flexibility in the batch application.
Thank you. We have surprisingly high number of questions regarding tracking capabilities which you did not show. So let me start with one. The question is there, this researcher studies um, embryos and wants to know, is there a way to analyze lineages?
Yep. So this is a little bit broad, and depending on your question, I might use certain tools and certain functionality into Avia, within Avia. So let me try to capture as many of the possibilities I can think of. So we do have a recipe that is specific to 2D tracking. So for example, if within your embryo you're doing 2D images and you're just trying to track the nucleus over the nucleus over time, that's possible with one of our recipes. That also comes with lineage tracing too. So you can see when the cells divide, there are markers to see when that division happens. And there's also a chart functionality that comes with it to see both the parent and the daughter when that division happens. So there's some capabilities there.
Awesome.
We can move. Sorry, Wanyan, I just wanted to cover the 3D portion too. I don't want to miss anybody who's potentially doing 3D. So for those who are doing 3D, there's also 3D tracking along with that too. So don't forget that you have that capability to do that. And I don't want to miss another a potential way for you to know. Not only can you track, because we're talking about embryo, not only can you track, but another thing that you might be interested in is taking a look at the whole embryo, counting the number of nucleus that's in there, or counting the number of vesicles or protein of interest that you have in this particular embryo. That's possible with our 3D cell analysis, and all of those hierarchies are built in for you so that the measurements of how many vesicles I have in this embryo, things like that, are also embedded into that recipe. So depending on what you're looking for, a couple of different features for you to use.
Thank you. We have a related question. Can we use this software to measure velocity of 3D cells?
Yep, so with the tracking, there are a number of measurements that are built in and by default calculated when you run the recipe. I believe it is one of it is the velocity. If it's not, and I'm wrong, you do have the option of a much larger list of measurements in our measurement tab. By default, not everything is calculated for you, but you can go in there. It's all pre-programmed into Avia. You just simply have to select the measurements that you want from the list, and then Avia will run those measurements for you. And velocity is definitely a part of the list of measurements.
Awesome. The next user sounds like a current user. This researcher asks, we use previous versions of neuron tracing in Avia to trace microvasculature. Is there any special version you plan that would specialize on microvasculature?
So at the moment, we do not have any plans to specifically launch a feature for microvasculature. But I'm sure as you might have tried, I don't know if you have, but if you haven't, do feel free to use the 3D analysis recipe with just the dendrite analysis portion to do the microvasculature. I've done that in the past and have gotten some pretty good results. For those who are here that don't know the details of the 3D analysis recipe, in this particular case, we did focus on detecting the soma and then tracing the dendrites. You do have the option in that recipe to detect only the dendrite portion. That might be of particular interest to those who are taking a look at microvasculature or specifically wanting to focus in on the dendrite and maybe combining that with a spine analysis. So that possibility is built into the recipe for you already.
Thank you. This might be the last question that we have. Is there a possibility to use own Python code to expand the functionality of Avia?
Absolutely. So Python integration has been a part of Avia for a number of releases here. So yes, you can utilize Python and add your code in. to further your analysis. We also have a GitHub that has a number of Python code and scripts already prepared for you from the customer success managers we have on the team. And you can drag and drop those and use it within Avia too. For those who may want to kind of utilize some of the Python scripting that we've done, but don't necessarily want to put their Python code together, we do have some things already packaged for you.
Thank you again, Quyen, for your time today and your important features that you presented. We would like to also thank Labroots for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today for their interesting questions. If you have additional questions, please feel free to contact us, and we'll absolutely answer every question that comes in. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who might have missed today's live event. Until next time, take care, everyone. Goodbye.